Currently, some scientists are researching a method called in vitro gametogenesis (IVG), which involves cultivating eggs from stem cells extracted from skin or blood cells. Hi, guys. I am Joanna. Happy to talk to you again. Today I am going to talk about something in the field of medical technology. Ready? Let's go. Since the birth of the world's first test-tube baby, Louise Brown, in Manchester, England, in 1978, IVF, that is in vitro fertilization, has been the primary treatment solution for infertility over the past 45 years. Despite the passage of so many years, IVF still has several shortcomings. Firstly, the process of egg retrieval remains a painful experience for many women. Moreover, the cost of undergoing a complete IVF procedure is a significant financial burden. In the United States, it costs around $20,000. Secondly, in many countries, traditional cultural values continue to influence attitudes toward this procedure. For example, some countries only allow heterosexual couples to apply for IVF, or they may prohibit the freezing of eggs. Most importantly, the success rate of IVF is not entirely satisfactory. After all, most couples seeking treatment have missed their optimal fertility age or have some underlying health issues. In countries like the United States and the United Kingdom, only about half of the couples undergoing IVF successfully bring their babies home. This data includes multiple attempts and years of trying. Currently, some scientists are researching a method called in vitro gametogenesis (IVG), which involves cultivating eggs from stem cells extracted from skin or blood cells. This process has been successfully achieved in mice, resulting in the cultivation of mouse embryos. Earlier this year, researchers claimed to have produced mouse pups using IVG, with one contributing sperm and the other contributing skin cells. The specific process involves extracting stem cells from the skin of a male mouse's tail. These stem cells are then genetically edited and developed into early-stage reproductive cells of an embryo. These reproductive cells are then mixed with supportive cells extracted from a mouse's ovaries, thus cultivating the precursor cells of eggs. These cultivated eggs are then combined with sperm from another male mouse. Finally, the fertilized embryos resulting from this combination are transplanted into a surrogate female mouse's body for gestation. Currently, only eggs have been successfully replicated using the method described above. However, there are very few successful cases of generating sperm using the same approach. In the natural world, the production of sperm requires the presence of the Y chromosome. Could this be the reason? In any case, if there is no further innovative discovery, the concept of reproduction involving two female bodies remains unachievable. The current research results are far from being applicable to human society. Additionally, this method poses significant ethical challenges to our existing understanding. As far as my knowledge goes, this is the extent of the information available. If there's more information, I'll be sure to share it with you. Okay, that's it. Bye-bye.